So we're going to talk about extracellular receptors, and the first type that we're going to talk about are the G protein coupled receptors, which we refer to as the GPCRs. It's a major superfamily of cell surface receptors in eukaryotes, and just a few important principles of the GPCRs. One is that it's a single polypeptide chain that spans the cell membrane 7 times. So these are kind of weaving in and out of the cell membrane like a snake. It's very serpentine-like. Just 7 transmembrane spanning domain. It's very characteristic of the GPCRs. Also, the name comes from the fact that these receptors interact with guanine nucleotide binding proteins. Okay, so that's where the G protein coupled part of the name comes from. So if we were to draw a cartoon of what this would look like, I could have a cell membrane like this, okay? And I'm gonna draw in a generic GPCR. So we said that it spans the membrane 7 times, and so we can see that, right? 1, 2, 3, 4, 5, 6, 7. And we can also see that the N terminus is on the outside of the cell and the C terminus is on the inside of the cell. Now, what happens is that the G protein, the guanine nucleotide binding protein that we're talking about here, is a heterotrimeric complex. And so we have a situation where we can just depict this as 3 subunits, a gamma, a beta, and we'll call this a G alpha, kind of connected to the membrane there. And the guanine nucleotide binding here is going to be the GDP bound to the alpha subunit, and we'll say that's in the inactive state, okay? Now, when the ligand comes and binds on the outside, that's why these are extracellular receptors. So we have some ligand here, okay? That's the molecule that's going to activate the receptor. You're going to get a conformational change in the receptor, and that's going to result in a change in affinity of the G alpha subunit for GDP. That goes down, and instead it's going to bind GTP. And so the result of that is going to be, I have my gamma and my beta subunits. They're going to be off to the side here, and this dissociation of the alpha subunit bound now to the GTP, and now this is active. Okay, so we have this heterotrimeric complex over here, heterotrimeric because we have 3 different subunits, an alpha, a beta, and a gamma, and we can see that when we have this separation and the GTP is bound to the alpha, that's really going to do the lion's share of the effects. And what's going to happen is that typically here, this is going to have an effect on a target enzyme that's going to result in what we call the signal transduction pathway, which we will discuss more specifically in additional videos. And so that is the way that you activate these G protein coupled receptors. I also will point out that the alpha and beta subunits that I have off to the side here, while I'm not really showing them doing anything in this particular example, there are cases where the gamma and the beta subunits can have effects as well, which we may talk about in other videos. But right now it's the alpha subunit really that's going to be having this effect on signal transduction. And so what has to happen in order for this pathway to turn off is that we need a mechanism that can bring this back to the inactive state where all the subunits are together and the alpha subunits bound the GDP. And we will talk about how those mechanisms work when we go into the individual pathways. So finally, the last thing that we'll say here is that in terms of the G proteins, there's three major types of G proteins that we'll focus on, and we call that GS, GI, and GQ. And yes, there are other G protein to think about, but these are the major ones involved in signaling that are going to be relevant to a lot of the effects of drugs and also various signaling molecules within the body.